Hello, everyone, and welcome to today's webinar, Chromatin Immunoprecipitation, 5 Steps to Great Results. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit www.thermofisher.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Eliza Small, Scientist 3, Protein Biology, Antibodies and Immunoassays, Thermo Fisher Scientific. Dr. Small has worked at Thermo Fisher Scientific since 2015. She works in the Antibody and Immunoassays Group and focuses particularly on identifying new epigenetic targets to develop antibodies and antibody validation. Prior to joining Thermo Fisher Scientific, Eliza earned her PhD at the University of Chicago, where she studied the mechanism of regulating the spliceosome by several pre-mRNA splicing factors. Eliza did her postdoctoral work at Northwestern University, examining nucleosome position at the single-cell level in yeast and the outcome of several epigenetic mutations in hematological malignancies. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Small, you may now begin your presentation.
Great. Thank you so much, Alexis, for the introduction, and thank you, everyone, for joining this webinar, whether live or later on demand. Today, I'm going to talk to you about the ins and outs of CHIP, or chromatin immunoprecipitation. As I was already introduced, my name is Eliza Small. I've been with Thermo Fisher for just over 3 years, where I work in the Antibody and Immunoassays Group, focusing on new product and development and validation in epigenetic and signaling research. Prior to that, I was a postdoc at Northwestern University. I started my postdoctoral training in nucleosome positioning in Jonathan Whittam's lab and continued that work, as well as examining several epigenetic factors in John Licht's lab. I did my PhD at the University of Chicago examining spliceosomes and how they are regulated in the Staley lab. So now on to talking about CHIP, or chromatin immunoprecipitation. CHIP is a technique for getting a snapshot or picture of protein-DNA interactions at a given moment. There are 2 main types of CHIP, which I will discuss in more detail in a bit, but they are Crosslink CHIP or X-CHIP and native CHIP. There's also a number of variations of CHIP that have been developed recently, and those, though, however, will not be discussed in this webinar. With this technique, you can ask where your protein of interest is and is not in the genome at a single time point. So you can ask if your protein that you're interested in is associating with actively transcribed genes, repressed genes, or is it in discrete regions in the genome, or spread out throughout the genome. Further, if you start beginning to study gene regulatory elements, you can actually start to obtain a 3D picture with this 2-dimensional data. There's a number of things to consider before starting the CHIP experiment. First is, what do you know about the binding of your protein of interest? Do you know that it binds DNA directly, or does it bind DNA as part of a protein complex? And depending upon how large that protein complex may depend on how you want to set up your experiment. Also, if the protein binds the DNA directly, how stable is that binding? Is it rather transient, coming on and off the DNA rather quickly, or once it binds, it's quite stable there? Other things to consider are, what are you interested in learning about your protein? Do you know definite regions that your protein of interest binds that you're interested in investigating? Or are you interested in understanding where your protein is on a more genome-wide level? Also, do you even know regions where your protein is generally expected to be bound and not bound? Another thing to think about before you start your CHIP experiment are all the reagents and equipment you need for CHIP. Not surprisingly, you need a place to grow your cells. You need water or heat blocks where temperature can be controlled. You, in most cases, need a sonicator. You'll need access to a qPCR machine for being able to analyze your DNA output from your CHIP experiment. And you will need a lot of enzymes, buffers, beads, antibodies, and other reagents. To that point, often CHIP kits are considered a great option, as many of the reagents required for your CHIP experiment are included in the CHIP kit. Here I show on the left a snapshot of a magnetic CHIP kit offered by Thermo Scientific, and you can see there's a bunch of reagents that are included in this kit. Beyond having a number of the reagents, they're all optimized for CHIP. That being said, you'll see throughout the webinar that CHIP requires lots of optimization for your cell type and what your protein of interest is. So although these kits are optimized for CHIP, you still will need to do plenty of optimization on your own for your specific experiment. I also want to mention at this point that on the right side there's featured products. So in this case, it's the 3 CHIP kits that Thermo Scientific has to offer. But throughout this webinar, I will often have on the right side featured products that I would recommend for your CHIP experiments. So now let's begin. In this webinar, it's divided into 5 sections for CHIP. Crosslinking and lysis, which is where you stabilize complexes and isolate nuclear fractions. Then the chromatin fragmentation, where you take that very long chromatin and fragment it so that you can get optimal resolution and efficient immunoprecipitation. The immunoprecipitation step is where you capture and isolate your protein DNA complexes. So this is where you're pulling out your protein of interest and finding out to ultimately find out what DNA it associates with. Then the preparation of DNA, and finally the quantitation of DNA, which is really what your output for your CHIP experiment is. So let's get started. The first step is crosslinking and lysis. So the first question is, do you need to crosslink your CHIP experiment? And that really depends on how stable the interaction you're studying is. If it's not stable, such as a transcription factor or another factor that you expect is coming on and off your DNA rather quickly, which is many factors in the cell, you really do need to crosslink, as you need to kind of freeze that protein to your DNA so that you can later pull it down. Alternatively, you might be interested in a very stable interaction. And this very stable interaction is something like a histone modification, where the nucleosome is pretty tightly on the chromatin, and so you may not need to require cross-linking at that. The question, though, is, in your CHIP experiment, are you only interested in these very specific stable interactions? And if that is the case, so if you're interested in, say, several histone modifications and how they associate with the genome, then native CHIP or N-CHIP may be the way to go. And the benefit to doing native CHIP is that cross-linking in itself does increase your background, so you've eliminated one issue in a CHIP experiment. However, often you're interested in not only that histone modification, but you're interested also in some other factors involved in one kind of overall CHIP experiment. So you might cross-link a bunch of cells and then divide them up and do several immunoprecipitations. And if you're interested in both stable and unstable interactions, you're going to want to cross-link. So again, it just depends on what you're interested in, if it's necessary to cross-link or not. Now, if you are cross-linking, the question is what is the right cross-linker? First and foremost, you're going to want to use formaldehyde. Formaldehyde is the most common cross-linker, and it cross-links protein and DNA interactions, although it does also cross-link very close proximity protein-protein interactions. It's reversible, which is key because ultimately you want to release the DNA from the protein. Now, if your protein of interest is a complex and does not directly bind to the DNA, you may need an additional Crosslinker. So at the bottom, if you were interested in, of course, the red or blue proteins, you would not need a Crosslinker. But if, say, you're interested in that gray protein that's depicted as a ball, as that's in a larger complex and further away from the DNA, you may need an additional Crosslinker, and DSG and DSP are two such Crosslinkers that are useful in this case. Now I'm going to go through quickly the kind of general workflow for crosslinking. In this example, it's using 1 million cells per mil, but this is something that can very much be scaled up or down, and people are doing lower and lower numbers of cells and having successful CHIP. Once you dilute your cells in the media to the appropriate concentration, if you need a protein-protein crosslinker, it'll be added at this stage. After that, you'll fix the cells with formaldehyde and incubate. Often for about 10 minutes is an appropriate length, but this is a step that may need to be optimized for your cell type. After the incubation, you want to quench with glycine, and this is really essential as over-crosslinking can later cause problems down the road in your CHIP experiments. You do want to quench, incubate that quench, and then wash off that glycine and formaldehyde with PBS. And this stage is one of the best stopping points in a CHIP experiment. It's the stage where you cross-linked, quenched, and washed your cells, and these can be stored at -80, the cell pellets, almost for several years at least. So it really is a great place for stopping your CHIP experiment if you don't want to continue, or if, say, you want to do some chromatin IPs at this stage, but also save some more cells later for doing another type of experiment. Next, after cross-linking the cells, or if there was no need to cross-link the cells, your cells would often have been washed in PBS and either stored at -80 or continuing right on, you need to lyse the cells. And lysing the cells, there's multiple ways to do it, but the basics are that you use a detergent for lysing or opening up the cells, and this allows you to get access, for your antibody later to get access to the nuclear fraction where your protein DNA complexes are. So the key to lysing, however you choose to do it though, is that you really want to see this morphological change. So shown here are HEC293 T cells, and on the left is a crude nuclear purification before lysing. So you can see these nice, with the DAPI staining, the nice nuclei. Now with just 3 minutes of nuclear lysis buffer followed by 30 seconds of sonication, you can see on the right a huge morphological change. And that's what's key, no matter how you choose to lyse, is that you see this large morphological change. Because if you do not properly lyse your cells, your CHIP experiment will not work, as the antibody cannot even get in to pull out that protein DNA interactions. Also, you'll likely even upstream of that have problems fragmenting your chromatin. So some tips from this step are, some that I've mentioned and some that I have not yet, are to spin the cells gently. I know I did not mention this yet, but this is something you want to make sure to do, because although you do want to lyse your cells, you don't want to prematurely lyse them. This is a step where you have to carefully lyse the cells, and spinning the cells gently is important not to lyse them. Crosslinking is time sensitive. Excessive or inadequate crosslinking can both lead to both increased background or noise and also a loss of DNA. Excessive or over crosslinking can also lead to chromatin that's difficult to fragment, which will reduce both your resolution and your IP efficiency. Different types of cells require different stringencies of cell lysis, so each cell type, the lysis step may need to be optimized. In some cases, just lysis buffer is adequate. In other cases, you'll also want to add a sonication step. You want to keep buffers cold and on ice during the whole time. And even if you're taking cells from the -80, you'll actually want to thaw those cell pellets on ice before you begin your lysis step. So that covers our chromatin, or our cross-linking and lysis step. And now we're going to move on to chromatin fragmentation. And this is where you're cutting up your chromatin to more manageable sizes for your experiment. So you can imagine you have this very long piece of DNA or chromatin with all of the proteins bound and cross-linked. And what you're interested ultimately in doing is finding out where your protein of interest is on the genome. And what's important there is to fragment your chromatin into manageable sizes so that you can actually get a good picture of where your protein is and is not on the genome. So to do that, you want to use some sort of fragmentation process, whether it's chemical or enzymatic. And the first option is, whether it's chemical or mechanical. The first option is mechanical, which is sonication. And this method is used for, again, fragmenting your chromatin. And what you want to do is, as you'll see on the right, at lower pulses, you kind of see this large smear of chromatin, and as you increase the pulses in your sonication, you start to get more and more small fragments of chromatin, and that's exactly what you want. The ideal range is between 200 and 800 base pairs, but I'd really say what you want is more like 200 to 300 base pair for fragmentation is really ideal, and especially if you're looking to continue your experiment to ChIP-seq or next generation sequencing, because then you can really get a nice footprint of where your protein is and is not on the genome. Sonication needs to be optimized for every cell line. It's something that's very important. You cannot rely on one method working for across the board of cells. It also needs to be optimized for every sonicator. So once you find a sonicator that works, you often want to continue to use that sonicator. It's important to keep the samples very cold because the heat can actually reverse the crosslinks at this stage, and then ultimately you'll have a very inefficient ChIP experiment. You need to prevent foaming when you're sonicating, as foaming will actually reduce the efficiency of the sonication. This method, although very tried and true, it's also a very hands-on method to fragment your chromatin. An alternative option that has become, I think, more popular is an enzymatic digestion, and specifically micrococcal nuclease, or MNase, is used for this. And what MNase does is it basically chews up any unprotected DNA, which really means it chews up most of the DNA that is not associated with a nucleosome. So I would say this method is particularly good if what you're interested in is looking at histones and histone modifications. A huge advantage to it is that you can perform multiple samples at the same time. And shown here on the left is a gel, and what you'll see is as you increase time with the MNase digestion, you start to see mono, di, and trinucleosomes. And dinucleosomes are kind of that perfect size for ChIP experiments. But on the right, you might be able to see that, again, cell line dependence is very important to optimize. Cell line 1, you start to see basically at the first time point, you can already see mononucleosomal DNA appear where it's really the second or third time point in cell line 2. And these samples were processed at exactly the same time with the same enzyme. So every cell line needs to be optimized. Additionally, the enzyme can start to lose activity due to age and or storage. So with MNase, you almost need to optimize for each experiment you do to find that perfect time point to get that mostly dinucleosomal range. MNase, although it does have a sequence preference, that's probably not as important with ChIP, but what is important to know is it really is chewing up all the DNA except for what's associated with the nucleosome. So it really has a nucleosome preference, I would say, in this case. So some tips from this section are, again, some I mentioned, some I've not. One that I know I did not mention is that you really want to add protease and phosphatase inhibitors to your dilution buffers. And why this is so important is that you do want to maintain all of those modifications on your proteins and also maintain just the general integrity of the protein. So again, if you're interested in histone modification, you want to make sure to maintain that throughout. Fragmentation conditions need to be optimized for each cell line, regardless of whether you're doing chemical or mechanical fragmentation. Always confirm that the fragmentation is in the desired range, because if your fragmentation, if it didn't work for whatever reason, then ultimately your resolution is going to be very, very minimal, as well as your ChIP efficiency will be incredibly low. For sonication, you want to keep samples cold and do more pulses at lower power. For the enzymatic digestion or MNase digestion, you can optimize time, concentration, and temperature to all try to get that right amount for getting mostly dinucleosomal size fragments. Sonication and nuclease digestion can actually be used in combination for particularly difficult cells to fragment. This is another stopping point, although I would say not as ideal because you usually do take a little bit of a hit on your protein, and so you are going to reduce your efficiency a little bit in freezing at this step, but it certainly is a place where you can stop. So now we've covered chromatin fragmentation, and next we're going to move to immunoprecipitation. And I will say this is going to be really the bulk of this webinar. Immunoprecipitation is where you are actually pulling out your protein of interest with an antibody and the DNA that it's associated with. Picking the right antibody is key for this, and there's three main classes of antibodies. The first is a monoclonal, and one advantage to it is that it has significant batch-to-batch consistency, particularly if you're using a recombinant monoclonal antibody, and recombinant antibodies are becoming more and more readily available. One potential disadvantage is that it recognizes a single epitope. So what that may mean for your protein is if it's in several conformations in the cell, your antibody may only recognize one of the conformations. So that's a disadvantage to monoclonal, and the advantage to polyclonal is that it recognizes multiple epitopes, so that potentially you're going to get much more efficient IP, and you're kind of getting a more of a better picture of where your protein is interacting in the genome, based on that the polyclonal antibody may recognize more conformations. The unfortunate part of polyclonal antibodies is they're a very limited resource, and there's differences from bleed to bleed in an animal, and then it's also dependent on that animal, so that they're a much more limited resource than a monoclonal. Thermo Scientific offers an oligoclonal antibody, which is actually a pool of our monoclonal recombinant antibodies. So I think that there could be some real advantage to that, as there's potential for it to recognize multiple epitopes, as it's this pool of monoclonal antibodies, and we do have absolutely the batch-to-batch consistency. Now, picking the right antibody is probably the utmost importance in your CHIP experiment. And the question really becomes is, how do you do that, and how do I know my antibody is specific? So the thing is that it verified in CHIP, what does that really mean? You want to see that an antibody is enriched in expected regions where you expect your protein to be in the genome, and depleted in other regions where you do not expect your protein to exist in the genome. That's in an ideal case. Sometimes you don't know that much information about your protein. Even a further good evidence, although again may not be possible, is that enrichment disappears or is reduced in a knockout or knockdown cell line in your CHIP experiment, or there's a change in enrichment upon cell treatment. So it's another way to really feel confident that your antibody is specific for your protein of interest in CHIP. I'm going to show some data on a histone-specific assay called SNAP.Chip that EpiCypher recently has launched, and also show some data on CHIP Westerns, which is another way to test specificity. And all of these are really focusing on CHIP specificity, because we're learning more and more that an antibody that's specific in another application, that specificity does not always translate to CHIP. Sometimes you cannot find an antibody that's been shown to work in CHIP, and one idea is to start looking for your own and testing it. And if that still isn't successful, affinity tagging is another alternative. The challenge, of course, is that first of all, you have to add the tag to your antibody or to your protein of interest. And additionally, beyond just adding it, then you have to worry about whether your tag has any effects, off-target effects on your protein. So I'm going to show now some data, both from our own website and produced by Thermo, as well as from some of our collaborators, showing some different ways we've verified antibodies in CHIP. So the first example is enrichment and depletion. So XPF is involved in DNA repair and is expected to be located at promoter regions. IgG on this graph in yellow is the no antibody control. So this is just the beads without the antibody. And so you do not expect to see any enrichment, which we do not see. So that's good to just show you're not just pulling down non-specifically on the beads. Now with the antibody XBF, indeed we're seeing a large increase in enrichment in these promoter regions, and then in the GAP-DH gene or satellite DNA, where we do not expect to see XBF, there is very little enrichment. So that's a nice showing of suggestion that indeed this antibody is specific. Another one shown here is H2B lysine 20 acetylation, which is associated with actively transcribed genes. And again, in this case, you can't even see the IgG or no antibody control because the full enrichment is so high. And you see a huge increase in active genes, whereas very little enrichment in these less active genes. One more example is H3K36 trimethyl. This modification is known to associate in gene bodies, and in this data, it shows 3 different genes, and you can see the promoter has much less enrichment compared to the gene bodies of gamma-actin, ALDOA, and GAP-DH. So this, again, is a nice demonstration of enrichment in the expected regions, in this case, the gene bodies, and much less in promoter regions. Now, I mentioned cell treatment as another way to test, or even a kind of, I would say, further specificity test, and this is shown here. So CREB, which is a transcription factor, is activated by phosphorylation. And on the Western blot on the left, you can see that with the addition of PMA, which is the second lane, PMA activates protein kinases and thus increases phosphorylation. And indeed, you can see it on the Western blot that there's an increase in the phospho-CREB in the second lane. Now, the chip on the right shows something similar. So in blue is that IgG, or no antibody control, for several different regions in the genome. And then you can see some enrichment in a couple of the regions when you are just the cells untreated. So that's the yellow. So particularly the second and third gene regions, you see some nice enrichment. Now when you add that PMA that has activated the protein kinases and increased phosphorylation, you see a huge enrichment. So that's showing a change in enrichment upon cell treatment, being a really nice demonstration of specificity. Now we're going to move onto a histone-specific validation method. And this is called SNAP.Chip and has recently been developed by EpiCypher. It originally comes from ICE-Chip, which is a method developed in Alex Ruthenberg's lab at the University of Chicago. And the way this method works is that they take recombinant nucleosomes, so they know what modification is on it, and there's the DNA wrapped around it has a barcode. And this panel of recombinant nucleosomes, in this case it's the K-METstat panel, that includes mono, di, and tri-nucleosomes for H3K4, K9, K27, K36, and H4K20, are spiked in to your normal chip workflow. And then after processing and you purify your DNA, you can see not only is your antibody pulling down the modification you're interested in, but is it pulling down any of the other modifications as well. And here's an example of data from one of our antibodies. So on the right is H3K4 trimethyl antibody. And what you can see, the black bar is the enrichment. So this is how the kind of efficiency of enrichment of CHIP, and it's really nice and high. And then what you want to see for a specific antibody is that you want to see that only it's really pulling down H3, in this case H3K4 trimethyl, that's what the antibody is targeted for, and very little of anything else. So this is exactly what you want to see for a nice CHIP-validated antibody. And I should mention that peptide arrays have kind of been the standard for histone modification specificity. And although they are fantastic for, I think, identifying looking at a large panel, they are a denaturing application and seem to be much more appropriate for Western blot. As a recent study in Molecular Cell by Alex Ruthenberg's lab, EpiCypher, and some of their other collaborators have shown that peptide array and SNAP.Chip, or in this case, ICE-chip efficiencies, specificities do not correlate. So an antibody that might look great by peptide array, that does not mean it looks great for specificity in CHIP. And that's why I think this method is so important for these histone modifications. So back to the data on this slide is that I was going to say that the ones that are starred, we've shown similarly to this H3K4 that it's really a clear, clean panel where it's only pulling down the target of interest and less than 20% cross-reactivity with anything else in the target. The ones in blue shaded boxes in the panel, we still have not yet identified a great antibody in our portfolio for CHIP, and that's very much an ongoing process, and hopefully we will have some of those very soon. And there's a couple that are not starred, but they're not shaded out, and those are ones where we're still trying to find, we have something, but it's not absolutely ideal. There might be a little bit higher than 20% cross-reactivity in at least one modification. Now the question is, as I mentioned, peptide array and ICE-chip or SNAP-chip specificity do not seem to correlate for histone modifications, but how important is this specificity? And the answer is it's very important. So this is a little bit of a complicated slide, and this actually comes from data from that molecular cell paper I just mentioned, so you can see the reference at the bottom. And what they found is, again, in the circle right at the top, there's this ICE-chip specificity. So for H3K4 monomethylation, you can see that, and that's in green, both antibodies they looked at by ChIP-seq have ICE-chip specificity of over 90%, and their ChIP-seq tracks are very similar. The same with the dimethyl, where the specificity is over 90%, and they have very similar ChIP-seq tracks. And I should mention that on the right, those numbers in black are actually our catalog numbers for these antibodies, the Thermo Fisher antibodies used in this study. Now, when we get to red, things fall apart, unfortunately. So this is the H3K4 trimethyl, and the top two red ones have only around 60% specificity, and they do have very similar ChIP-seq tracks. But when you look at the bottom two red ones that are more hovering around 90% specificity, they have significantly different ChIP-seq tracks than their counterparts above. And what those extra peaks in the top two red ones of H3K4 trimethyl are likely coming from is the dimethyl. So this is really skewing your data, where when you think this is where H3K4 trimethyl is, it might actually be the dimethyl. So having this kind of method is essential for understanding your ChIP, particularly if you're going on to ChIP-seq, but even in ChIP-qPCR, your data could be confused without this specificity. Now, this is a very histone-specific technique, so we're also interested in plenty of antibodies or targets that are not histones. So ChIP-Western is a good way to also look at specificity. This is another method that kind of can be incorporated into your normal ChIP workflow. In this case, you crosslink your cells, lyse the cells, fragment the chromatin as you've done before, and do your immunoprecipitation. But instead of then going on to purify the DNA, or at least taking a fraction of this immunoprecipitation, you examine not just the DNA, but the protein. So you boil the protein off the beads, and then you can do a Western blot with the same or different antibody to the target used for ChIP, and see, am I pulling down my protein of interest? I'm going to show a couple examples of that, the first being lamins. And the reason lamins are hard is that they provide the nuclear structural organization, but they're also all over the place, so it's challenging to determine antibody specificity, as it's hard to identify regions of depletion. So on the left is lamin AC, and what you'll see in the first lane is your input, so this is what you're putting into your immunoprecipitation. The second lane is just IgG, so no antibody, and then the third lane is the actual IP, and there again, so in the input, you would expect to see your protein, as well as in the lane 3, your IP. Similar story on the right, and this is looking at lamin B1, and this actually looks at two antibodies from other suppliers in lanes 1 and 4, and two Thermo Scientific antibodies in lanes 2 and 3. And all 4 antibodies are showing that they are indeed pulling down lamin B1, suggesting that this is your CHIP experiment is working and it's pulling down your protein of interest. Another example, this is for an RNA-binding protein, so probably would be more appropriate to call it a Northwestern. And this is for RNA Pol2 on the left and ELAVL1 on the right, which binds and stabilizes mRNA. And in this case, I'd recommend focusing on lanes 2 and 4. So lane 2 is the pull down of IgG, so no antibody, and in both cases, or in all cases, you would expect to see no protein, as you are not actually adding antibody, and indeed that's the case. Now lane 4 is where you do your actual IP, and in both cases you see protein, and in some cases you'll actually see it enriched over your inputs, which are in lanes 1 and 3. One final example of CHIP Western actually includes peptide competition, which is an even kind of additional specificity test. So in this case, again, you have your input lane and then your IgG or no antibody. So, and then you add in either no blocking peptide or blocking peptide. This is for antibody H3K36ME3, which I showed you some data earlier that it associates with gene bodies. And we've also validated this antibody in SNAP.Chip and looks really nice. In this case, you've either had no peptide or a K36 mono, di, or tripeptide. And in the case of no peptide or mono or dipeptide, you see H3K36ME3 as labeled, and then that band disappears in that last lane where the K36 tripeptide has been added. And further, if you look at the bottom of the gel, you actually see that peptide light up. So these are just all ways of looking at your antibody specificity and is my antibody really pulling down what I think it is? And then in some cases also, not only is it pulling down what I think it is, but ensuring it's not pulling down what you do not want it to. Now, another thing you may need to do is actually look at your antibody concentration. So once you've identified a good antibody, it might require some titering of your antibody to find the ideal amount. In this top example here, this is for an H3K27 trimethyl antibody. You can see as you increase from left to right the amount of antibody, you also increase the percent enrichment. This, however, is not always the case that more is better. The next two examples were provided kindly by EpiCypher. In the middle example, this is for H3K9 dimethyl antibody. And this time, the increasing goes from right to left. And indeed, if you focus on that black bar, you see an increase in enrichment as you increase the antibody. So that looks great. And you really see about similar, or even sometimes an increase in specificity, which is really nice because this is not just looking at H3K9 dimethyl pull down, but also looking at H3K9 mono, tri, and just H3. So this looks really nice, but you can't always count on that because in the bottom example, again, it goes from right to left on increasing the antibody. You do see an increase in the efficiency as you focus on that black bar, but for this one, it's an H3K4 trimethyl antibody, and as you increase the antibody, you also start to see an increase in the pulling down of H3K4 dimethyl, which is not what you want. So although there's an increase in efficiency of pull down, you're also decreasing your specificity. So the antibody concentration can definitely be a touchy thing. Usually you'll incubate your lysate with your antibody overnight, and then in the morning add beads. There's two types of beads you can use, magnetic or agarose. Agarose beads certainly have the advantage of higher binding capacity, but generally speaking, magnetic beads are a little bit easier to use, but it's really about what your comfort level is and which ones you have a preference for in most cases. You do need to select the right binding protein for the IgG antibody cell class. In most cases, protein AG is a great choice, but it is something you want to be aware of with your antibody, making sure that that is the appropriate choice. You just want to ensure your beads never dry out, and I would recommend vortexing very lightly to resuspend so you get a nice slurry before you add them. The next step after bead incubation is washing the beads. And this is a bit of a tedious step, but a very, very important step. So I highly recommend being very patient with your washing. You go from low to high salt washes, and you want to be careful with how the stringency. So in some cases, when you have these really strong interactions, you can really go quite high in your salt washes to really wash away the non-specific, whereas in other cases, you might want to go a little bit lower on the salt, particularly if you did not cross-link your cells. Then you'll do a lithium chloride wash, and that reduces the sodium and also switches the salt, which can remove other non-specific binding. You'll want to do a TE wash so that you get it kind of neutralized and ready for the next stages of DNA purification. So some tips from this section are picking the right antibody for CHIP is essential, and that probably would be the biggest take-home message I'd like you to take from this whole webinar. Validated in CHIP or IP antibodies is obviously preferable, and really being able to show specificity. And specifically, you want to show that specificity for CHIP, as not all application specificities are transferable. Sometimes, however, you're interested in something where there is not a clear CHIP antibody out there. IHC and IF are certainly good indicators that the antibody is a good candidate for CHIP, but that absolutely needs to be tested. You do always want to add a no antibody control to distinguish CHIP signal from background. Antibody titrations might be necessary for identifying the best signal-to-noise ratio. I would generally say 2 micrograms is a good starting point, or what the data product sheet for your antibody recommends is also a very good starting point. Washing stringency also may need to be optimized for your particular CHIP reaction and your particular antibody. So now we've covered the immunoprecipitation step, and the next step will be much quicker, which is preparation of DNA. And this is where you're purifying your DNA so you can ultimately see where your protein is associated. So for DNA preparation, you elute the chromatin from the antibody and then reverse the crosslinks as well as degrade the protein with proteinase K. RNase treat to remove all the RNA from your sample and then purify the DNA, either by phenylchloroform extraction and or a PCR cleanup kit. And that's really how you prepare your DNA. And then this is also another stopping point where your DNA can be stored at -20 for as long as you please. So that's the preparation of DNA. And now we get to the quantitation of DNA. So this is where you're figuring out how much is enriched, is enriched at any specific loci you're interested in. So the quantitation of DNA, you need to identify regions to examine. So what regions are you interested in? Do you know regions where you expect your protein to be and not be in the genome? And you want to design and test primers for those regions. And I would highly recommend actually designing at least a couple primer sets for each region, as its primer design and testing can be a little bit of a hit or miss experiment in itself. Then you'll do 2 PCR to actually amplify those regions and analyze that data. Now, this might be your stopping point. This might be you know what regions you're interested in and you do them and test them. Or you may now prepare your DNA for next generation sequencing or for ChIP-seq. So identifying the regions to examine. So if you're interested in where this blue ball protein is, you would identify a couple of regions and design primers to amplify these regions to see, is my protein there, or has my ChIP pulled down this region of DNA, and how much of it? You'll also want to design at least a region, if not a couple, where you do not expect your protein. Because what you want to ensure is that you haven't just kind of pulled down generally all of chromatin and you're amplifying everything. After you've identified those regions, you'll design primers. Amplification regions should usually be between 100 and 250 base pairs. I would err on kind of a goal of around 150 base pairs. Primers should ideally be 18 to 25 base pairs with a 40 to 60% GC content if possible. And that can be a hard thing sometimes to, I get that, depending on the region you're looking for. The TM should be between 50 and 65 degrees, definitely no higher than 65. And again, this is where I'd err towards kind of 55%, 55 degrees being a goal. You want to see a single dominant peak when you look at your melting curve, and I've just shown two examples on the here, where on the left is the good melting curve, and you see this nice, strong one peak, compared to on the right, the bad melting curve, where there's just lots of peaks. And then on the next slide, I'm going to discuss more about this, but you want to look at your primer efficiency, which should be between 90 and 105 degrees. Now, your particular qPCR machine may do this calculation for you, but this is showing you how to figure out primer efficiency and also just how to set it up. So you're going to want to take either your input from your chip reaction, or you can just use some genomic DNA in the freezer and do a qPCR where you've diluted the sample at several logs, and then that's going to be graphed versus the CT. And what the CT is it's the value or the cycle where your fluorescence signal in your qPCR reaches a threshold above background. And so that's kind of the raw data that your qPCR provides. When that's graphed, you'll get a slope, and you use that for this equation at the bottom for percent efficiency. And here's again two examples. On the left is the good primer efficiency. This is 100%. And then on the right, I mean, you can just see from the data, it does not look nice. All those blue dots are nowhere near the line. But this is a bad primer efficiency at 170%. Now, it might seem odd how it's actually more efficient, but what that means is it's actually amplifying not just the region you're interested in, but probably other regions in the genome. Now, the next slide I'm going to show you is a bit complicated, but provides the calculations for what to do with the data from your qPCR. In some cases, your machine might do this for you, but I think it's really important to understand what the analysis is. So there's two ways to graph your chip data. The first graph, which is shown already here on the left, is the fold enrichment, which just looks at the signal over the background or no antibody. And the equations are just above the graph. So your delta CT gives the signal minus the background. And so what you'll see is you take your IP signal, so this is, again, that cycle threshold, and then you minus it from your IgG or your no antibody control, and that gives you the delta CT. And you take this delta CT, and again, the equation is right above the graph to figure out the fold enrichment. And so again, this gives you how much is enriched above background in your experiment. Now, an alternative calculation is to look at the percentage of input. And what this calculation represents is the amount of DNA pulled down relative to the starting material or input. So in this case, first you need to adjust the input. So in general, your input, of course you're not taking 100% of your input as a sample, because then you would have nothing to IP. So you often take 1% or so of your input, which is what's done in this example. And you need to adjust this because 1% was used, only 1% was used for chips. So you take a dilution factor is 100, and the log base set of 2 of 100 is 6.65. So that's just to understand where that minus 6.65 comes from. So you take your input and then minus it by 6.65 to get your adjusted input. Then from there, you can calculate your percent IP. And again, the formulas are just above the bar graph. And here you take your adjusted input and minus it by the IP to get your percent input. So again, these are just two different ways to look at your data. And I know this is a little bit of a beast of a slide, but it's just to understand how those calculations, particularly if your qPCR machine just spits them out, just having a little background to how these actually are calculated. Now, that may have been your stopping point. You may have looked at a number of regions that you're interested in, and you figured out what you want to know about your protein, but you may already have known this, or you may have decided, you know what, I want to know much more about what's going on in the whole genome with my protein of interest. And here's where you'd move to NGS. And I'm really not going to talk about NGS, but just wanted to show you that the chip workflow here is shown on the left, and then on the right, you just kind of that workflow can go right into preparing libraries for ChIP-seq. So you're already well on your way to a ChIP-seq experiment. If you are planning right from the beginning of doing a ChIP-seq experiment, I still very much would suggest testing a few regions by qPCR to ensure that your ChIP experiment is good before you invest the time and money in sequencing. So a few steps from this section. Primers need to be assessed by their melting curves and their efficiencies on input or genomic DNA. A few important controls that I did not mention for qPCR is you want to have a no antibody, a no DNA, and a no reverse transcriptase control, just to make sure all your reagents are nice and clean and there's no contamination, as that would skew your data. And I've already mentioned you always want to perform a few qPCR reactions, even if your intention is to move to NGS, to ensure your ChIP is a quality experiment that you'd like to move to NGS. So some conclusions from this whole webinar are determining the goal for your experiment is key before beginning to design the best experiment. Things to determine before you start ChIP are the conditions you want to fragment your chromatin, identifying antibodies you want to use that you feel confident are good, or how you're going to test that they're good and specific, because that's sometimes what's necessary. And you also, I would recommend actually having your qPCR primers already ready to go and have them designed and confirming their efficiencies are good before you get too far in your ChIP experiment. As you've seen throughout, there's many steps in ChIP, and there's many places that need optimization for whatever your experiment requires, whether it's out due to the cell line or the type of target. There's lots of places that require optimization, and anywhere along the way that you can check to see if it's working, I highly recommend that. So things like checking to confirm that your fragmentation is good, and checking to confirm your lysis is good, and checking that your qPCR primers are good are all ways to just ensure along the way that you're getting the best experiment. And with that, I'd like to thank a number of my colleagues at Thermo Fisher Scientific who helped put this webinar together, as well as providing data for this webinar. Additionally, I had several collaborators kindly give me data to include in this webinar, and I thank them very much for that. And lastly, I thank EpiCypher, who shared not only data provided from their own work, but also have been great collaborators along the way. And with that, I thank you, and I will take questions.
Thank you, Dr. Small, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, ChIP Western seemed to show that the antibody is pulling down the protein of interest, but how does it show specificity?
That's a great point, because what I showed you is that you can see that your protein is being pulled down, but you have no idea what else it's being pulled down. In the case of the histone modification, it gave a little bit more information because the peptide competition was added, so that you could see then that it was competed off only when the trimethyl peptide was added. But you're absolutely correct that that doesn't fully show specificity. So things I would recommend doing are a Ponzu staining of your membrane, or silver staining of your gel. And what you would expect to see is mostly just your protein. You may see some other proteins, especially if your protein you know is in a complex, but that's going to add another layer of specificity by being able to just see a pretty clean gel or membrane where you're not seeing that protein.
Now, how do I identify where the non-specific binding is coming from, and therefore how to troubleshoot it?
Well, that's a hard question, because you probably saw throughout there's many reasons. So the first thing is to confirm that your antibody is really specific. And if you're confident that your antibody is specific, then that it's not coming directly from your antibody, then there are definitely things you can do. Unfortunately, if you're not confident your antibody is specific, or you find your antibody is not specific, then you just need to start over. But if you feel that your antibody is good, one place to look at is chromatin fragmentation. So you want to make sure, again, that you're on that kind of 200, 400, maybe a little above base pairs, because a lack of good chromatin fragmentation, you basically not only is your efficiency of pull down lower, but you've really kind of increased how much you can amplify in your DNA of where the protein is, so that footprint becomes much bigger, and then you're not getting that same nice resolution. So it might be that it's not pulling down something nonspecific, but that it's more that you're only seeing you're seeing kind of these large fragments that you're amplifying. Another thing would be, I'd highly recommend also using methanol-free formaldehyde, because methanol contamination can cause problems with fragmentation. Another thing that can cause problems with fragmentation is over-crosslinking. So if you've cross-linked for too long, it makes it really hard for fragmentation to occur. So all of those kind of things will make it look like maybe more nonspecific binding. Additionally, for nonspecific binding, you can alter your washing conditions, because that can lead to other problems, or that you haven't washed off all of the nonspecific. But yeah, it's a hard question, because there's just a lot of places where nonspecific can take place. But I'd say fragmentation and washing are some of the big ones if you're confident your antibody is specific.
We've gotten so many good questions today, but it looks like we only have time for one more. So our next question and final question will be, if my antibody works in the ChIP-qPCR, will it work in ChIP-seq?
If your antibody is really specific but the efficiency is low, sometimes that's okay, particularly if you're just doing ChIP-qPCR and it's kind of at these regions that you really know are going to be enriched. But what happens sometimes is, in that case where it's really enriched in certain regions, because that's kind of where all the protein is hanging out, then you find that when you do ChIP-seq, the signal, like the noise is really high compared to signal. So I would say mostly yes, your ChIP, if an antibody works for ChIP-qPCR, it should work for ChIP-seq. But the cases where it becomes more questionable is when your ChIP efficiency is low, and I would say kind of like hovering around 1-2%, I'd start to get a little more concerned that it might not work as well. And definitely if it's anything less than 1%, I'd be very nervous doing ChIP-seq with that. And then I would lastly say, probably ideally you want it probably 5% or higher is kind of, you're in a much more safe zone and much more likely to have successful ChIP-seq.
Thank you, Dr. Small. Do you have any final comments for our audience?
I would just say, there's lots of checks along the way, so really try to slow down and try to optimize every step where you can. And the other thing would be choosing the right antibody and really testing its specificity as stringently as you can for ensuring a successful ChIP experiment. And good luck.
Thank you again, Dr. Small, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand through April of 2019. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.